Every cell contains thousands of genes, but at any particular moment, only some of those genes are active. So how can scientists discover which genes are being used and which RNA transcripts are being produced? The answer is transcriptome sequencing. The genome contains the complete genetic information of an organism. The transcriptome is different. It represents all the RNA molecules expressed in a particular cell, tissue, developmental stage, or environmental condition. The process begins by collecting a biological sample, such as a plant leaf. RNA is extracted and prepared as a sequencing library. Depending on the technology, RNA may be converted into complementary DNA or sequenced directly. The resulting sequences are then analyzed using bioinformatics. In Illumina RNA sequencing, RNA is usually converted into complementary DNA and broken into small fragments. These fragments are sequenced in parallel, producing millions of highly accurate short reads, commonly between 75 and 150 bases from each end. Illumina sequencing provides high accuracy, very high throughput, and a relatively low cost per sample. It is especially useful for measuring gene expression and comparing large numbers of biological samples, treatments, tissues, or developmental stages. However, short reads usually represent only small parts of a transcript. When one gene produces several transcript isoforms through alternative splicing, it can be difficult to determine which complete isoform produced each short fragment. Transcript structures must therefore be reconstructed computationally. Long-read sequencing helps solve this problem by reading complete or nearly complete RNA molecules. Instead of assembling many small fragments, researchers can observe the structure of an entire transcript in a single read. PacBio Iso-Seq sequences full-length complementary DNA molecules. Repeated sequencing of the same circular molecule produces highly accurate hi-fi reads. This makes PacBio particularly powerful for discovering novel isoforms, identifying alternative splice sites, and improving genome annotation. However, PacBio Iso-Seq requires high-quality RNA and complementary DNA library preparation. It generally produces fewer reads than Illumina and may cost more per sample. It is therefore most valuable when accurate full-length transcript structures are the main objective. Oxford Nanopore can sequence long complementary DNA molecules, but it can also sequence native RNA directly. During direct RNA sequencing, an RNA molecule passes through a nanopore, and changes in electrical current are translated into sequence information. Nanopore sequencing can generate very long reads, detect full-length isoforms, estimate poly-A tail length, and retain information associated with RNA modifications. It also provides data in real time. However, its raw read accuracy may be lower than PacBio Hi-Fi, and direct RNA sequencing often produces fewer usable reads. Use Illumina when accurate gene expression measurement, high throughput, and large sample numbers are most important. Use PacBio Iso-Seq when you need highly accurate full-length transcripts, novel isoforms, or improved gene annotation. Use Oxford Nanopore when you need very long reads, flexible real-time sequencing, direct RNA analysis, or information about RNA modifications. In many transcriptome studies, the strongest approach is to combine technologies. Long reads can first identify complete transcript structures and novel isoforms. Illumina short reads can then quantify those transcripts accurately across many biological samples. Short reads tell us how much RNA is expressed. Long reads show us which complete transcripts are present. Different technologies provide different strengths, but they share one goal: helping us understand how genes function. Choose the biological question first, and then choose the sequencing technology.